this is physiology, which is, and it's human physiology, which is the study of the function of the human body. But just so you know, you're not, I know your mom loves you, but you're not that special. Your physiology is like the same as your cat's physiology, really. So we will talk about mammalian physiology, and my background is in mammals and evolutionary biology, and I use a lot of that kind of examples as well. So this is the study of the function of the human body. What makes it slightly more humanish is that people care quite a bit more about diabetes in humans than they do in cats or guinea pigs, right? So we sprinkle a little pathophysiology in to make it relevant, right? And so that you can see the clinical significance of a body process working properly. Does that make sense? So we'll do a little bit of pathophysiology. Now, the pathophysiology is not because I'm trying to teach you a patho class, but just because it makes physiology a lot more interesting, right? So a lot of times what we will do is with phys, I'll try to build a really good framework so that you understand how a body system is supposed to work. And then if you really understand it, what will happen is I can say, well, that part's broken, and then you predict how that would impact the physiology of the body system, right? And then it solidifies your understanding of normal functionality to understand what would happen if you broke it. Does that make sense? So that's really how I infuse patho into it. So, so here is how phys is different than anatomy. Anatomy was mostly memorization, right? Not very much that was conceptually difficult, right? It wasn't conceptually difficult that the forearm muscles were there, right? But those damn you didn't like learning them just because it was hard and there were so many things that you had to learn at one time. Phys is far less memorization and far more processing of the information that you get. Does that make sense to you? So there's a lot more thinking that goes on in phys than did go on in anatomy. Now, I'm not saying anatomy is easy. It's just a different kind of hard. So most of the time you will not feel like somebody piled information on. You will feel like somebody told you a really complicated story about how to put together a car and then let you leave. And now you're supposed to tell somebody else how to put together a car. And you're like, I'm pretty sure that there was something that was turning. And if it turned the wrong way, it was bad. But eventually, it starts to get in there. So it's less information, but more complexity. So here's what I do. With physiology, there are patterns that you can see in physiology that start to repeat over time. So like negative feedback interactions, if this gets really high, then it'll turn this thing down. And so what happens is what I try to do is I try to show you a pattern and then another iteration of the pattern and then another iteration of the pattern. But I'll just show you the first three. With the next four, I want you to recognize the pattern when it occurs and say, oh, I see, that's an example of negative feedback. Just like the one that we saw there and there and there, right? Does that make sense? So eventually what you get is you start recognizing the patterns and recognizing the connections, and then you start to get a big picture, and this big picture gives you a really strong framework, and you're like, okay, so this is what I think about the cardiovascular system. I think the cardiovascular system is all about pressure changes making blood flow, right? Okay, so it flows from high pressure, low pressure, and I'm trying to do that. And then I tell you, well, basically there's three ways to regulate pressure. You can make the tubes bigger or smaller, or you can make the pump harder, right? Or you can put more fluid into the system. You guys with me? Okay, now, and then I can tell you a couple of hormones that mess with those things. Do you think that I will tell you all of the hormones that mess with pressure? Or all of the drugs that you could possibly take that mess with pressure? Or that at some point I could ask you what a diuretic would do to blood pressure once you have the pattern down. You guys with me? So what does a diuretic do? Not to pressure, just in general. Yeah, it makes you pee, right? And therefore it decreases blood pressure because it messes with the fluid volume. So do you see? There's no way in hell that A, I could know everything about physiology, or B, that I could teach it to you. But what I can do is get you a really strong framework so that the next time you get a new piece of information, you know exactly where to put it. Does that make sense to you? So you get a really strong framework and you're like, okay, I got this. This is all about pressure. And this is just another example of something I've seen 27 times. And you will learn more examples than I could ever teach you throughout your career if you continue to go into clinically related stuff. And you will know more about drugs than I ever know by the time you're in your specialty, right? Okay, so I want to give you the information in a strong enough framework to categorize new facts, so that I don't have to tell you every single time that it exists. If I say it works like that, you're like, oh, then I predict it would do this, like every other thing that works like that. So you're going to be required to think a lot. You're going to be required to test your knowledge. You're going to be required to go out on a limb a little bit and hypothesize what you think would happen. And sometimes you'll be wrong, but by the time we get to the end of the semester, you're going to be right most of the time because you're going to have a really strong framework for predicting what would go on. That's what Phys is to me. Phys is not memorization. Phys is getting a strong enough understanding that you are really confident about the predictions that you're going to make. Does that make sense? Okay, so now, first thing that we are going to talk about is Phys is a lot of moving things from one place to the other, okay? And the things we will talk about and the places we will talk about, but an awful lot of human functionality, for instance, is getting things from the external environment to the internal environment and getting other things back out to the external environment. So a lot of times you know like the what that we're trying to get in or out, and you've known it for a while, right? You know that when we breathe, we need to get oxygen in, for instance, right? And that is no surprise to any of you guys. But what you might not know yet is how you get it in. What transport mechanism do you use to get oxygen to cross the respiratory epithelium? And why do you use that transport mechanism? And what would happen if it didn't work? That's the Phys, right? And then when you start talking about it more broadly, you can start talking about where things are exchanged, how they're exchanged, and why they're exchanged. So if we go to this figure, this is not obviously a human body because nobody looks like that, but this is a physiological drawing of the human body that represents the primary sites of exchange. So what I want to do is just briefly talk about all of the places that you are going to do exchange to do this one physiological process that we all understand. You eat a Snickers bar, yes? You're going to eat a Snickers bar and you're going to take the nutrients. Let's focus on glucose from that Snickers bar and get those nutrients all the way inside the cell of your little finger, the glucose for cellular respiration. Does that make sense? So what kinds of Phys is involved there and what kinds of questions will you be able to answer confidently by the end of the semester? Well, first off, we of course know this is the GI tract right here, right? Okay, so of course that's where you're going to take it in. That's no mystery to anyone. But the first question that we will have to answer is, by what mechanism do you absorb glucose across the intestinal epithelium? So what do I mean? I mean, how do you decide whether you get to diffuse it or use osmosis or use a channel or use a carrier or use ATP or don't, right? And it's not memorization. By the time you get a few examples, you'll realize that it's actually a little logic problem, right? The body is really lazy and cheap. It will do things the cheapest, fastest way it possibly can. So we're going to get it, let's say we get it in, okay? And then it's going to go into this extracellular interstitial fluid. But then we have another transport problem, don't we, right? We got to get it into the capillary, right? So the next question is, how do you get it in here? And then, of course, we are going to transport it. What would happen if you didn't transport it? If your cardiovascular system wasn't functioning, of course, the glucose would not get to the little cell, right? Because it's too far for diffusion. And then, of course, we're going to have to get it back out. These are the cells of your little finger that I'm trying to get through. This is a type of transport that you're going to have to predict. And then ultimately, you're going to have to predict how to get it across the cell membrane and what would happen if you didn't. For instance, what if I didn't have any insulin right there? Could I get glucose to move inside that cell in the absence of insulin? Very rarely can you get enough glucose to move inside the cell. So you will see that what happens is the whole Phys story is a bunch of exchanges, and then eventually you start thinking about how do I exchange it and what would happen if I didn't, right? That makes sense? So there's a bunch of predictions. And by the time you're done with Phys, you end up being really confident about these things. And so you start thinking about, okay, so now I got glucose in, maybe I got oxygen. Well, I know I'm going to generate carbon dioxide in these cells, and how does it get out, and what would happen if it didn't get out, right? That makes sense? So all of those things that you learned in 1610 when you learned about channels and carriers and osmosis and diffusion and all that stuff that doesn't seem relevant to anything. What ends up happening in phys, and one of the reasons I love teaching phys is a lot of those aha moments that made you figure out why the hell you had to take all those other classes. You're like, oh my God, that's why I had to learn pH. Now I care, right? Oh my gosh, we're talking about osmosis in the kidneys. I can't believe it. It's actually relevant to something. And then you put together with the anatomy, and then you're like, oh my God, it's one story. It's one story, just one, not 5 different stories that are completely unrelated. Okay, so we are going to do a lot of exchange of materials. Now, for those of you who have had anatomy, it should make sense to you that if you ever want anything to get inside the human body, it has to cross an epithelial tissue, correct? Because the epithelial tissues represent the boundaries of the human body. So here are your boundaries, and the primary places that you do exchange between the internal and the external environment are the GI tract, right? The respiratory tract, and the urogenital tract, right? Those are the 3 primary. Now, I'm not saying you do 0 through your skin, but certainly far less through your skin. Now, I want you to think about the clinical significance of having these places in which I am supposed to do transport, right? If you have a place that you're supposed to do transport, then you can't shut it off. And if you can't shut it off, it becomes clinically important because that's the place that you're most likely to get exposed to pathogens as well. Does that make sense? Aren't these the body systems, the respiratory, digestive, and urogenital, aren't those the body systems where you get exposed to most of your pathogens, right? So they're clinically important for 2 different ways, both in pathophysiology and in normal physiology, the GI tract, the respiratory tract, and the urogenital tract. So once I get it in to my system, then I have to get it across the cell membrane. FYI, just so you know, it's actually not considered inside your system until it crosses an epithelial membrane. So if you had a sibling that tortured you like mine did, I'm the youngest, and my brother used to feed me coins just to see how big a coin I could swallow, right? And I cannot swallow a $50, apparently. But I could swallow a quarter. But was the quarter ever actually inside my body? From a physiological perspective, it wasn't, because it came out just a dirty looking quarter out the other end. I promise I was 4. It's not that gross. So just came out a dirty looking quarter. So if it doesn't cross an epithelial membrane, it's actually not considered inside the body. Does that make sense? Okay, but once you get it inside the body, then of course you're going to have to get it across the cell membrane as well. Okay, so now let's talk about how things move in the human body and what they move through. Obviously, it's easier to move things through fluids than it is through solids, right? And you have heard 1,000,000 times that your body is mostly water. You are about 60% water, okay? And the whole collective water in your body is called total body water. And the total body water, notice, is in blue here, and it includes everything but the insides of those tubes, which are actually considered the outside of the body. And it would include all of the fluids that are inside your cells and also all of the fluids that are between your cells. Does that make sense to you? And so collectively, you have, if you're about 150 pounds, you have about 42 liters. That's a lot of water. You are mostly juice, right? 42 liters of water. So the first thing that we have to do is talk about where that water is sort of confined, and then we'll talk about exchange between them. So, first thing is that you start with your total body water, and then you start talking about how the total body water is separated. Okay, so total body water is just all of the water that you have. And then the immediate subdivision of the total body water is the ECF and the ICF. Now, when you were in 1610, they probably called ICF cytoplasm or cytosol or something like that. In phys, for some reason, they never use that term. They always just call it ICF. And ICF stands for intracellular fluid. Now, I know you've learned this 1,000,000 times, but I have to remind you, what does intra mean? Within or inside. And it's different from inter. And we're going to run into inter in just a second. So we have intracellular fluid. You have 100 trillion cells in the average size body, each of which has fluid on the inside. So 100 trillion pools, individual pools of intracellular fluid. And then the extracellular fluid is ECF, extracellular fluid. And what does extra mean? Outside, exactly. But the extracellular fluid has a further subdivision. And so collectively, so this is what your ICF looks like, all of the blue here, 100 trillion individual pools. This is collectively what your ECF looks like, right? All of the stuff that is fluid inside the body but not inside the cells. And then the ECF has a further subdivision, and the further subdivision is, what do you call the fluid that specifically surrounds your blood cells? Plasma, yes. And what do you call all of the other fluid? I don't know if you know this term. It's called interstitial fluid. What does the prefix inter mean? Inter. What? It means between, right? Interstitial fluid is the fluid between the cells. Now, in anatomy, when you took histology, which I know is everybody's favorite part of anatomy, remember looking at luceralar connective tissue? And luceralar connective tissue had cell, cell, fiber, fiber. And the stuff that wasn't cells and wasn't fibers was that, interstitial fluid. It was in the matrix, right? And it was fluid. That's why you could get things in and out of the tissue. Does that make sense? So that's interstitial fluid. Good? Okay, so now let's go back to the idea of transport. Primarily what we are doing is we are doing transport through these fluids in the body, right? And so I want you to think anatomically for a second, and I want you to think about the structure that I would have to cross if I were to try to get something to go from the blood plasma to the interstitial fluid. What anatomical or physical structure would I have to cross? That one's not exactly the plasma membrane.
A junction.
What?
Like a junction.
You could go through a junction. I want you to think about a capillary for a second. What would you have to cross to get the blood plasma? Something from the blood plasma? Yeah, the endothelium, the capillary endothelium, which is basically simple squamous epithelium, right? It's epithelium, right? And it's the thinnest of the epithelia, right? The simple squamous epithelium. And it can have greater and lesser degrees of porosity. But it does have some control over what's transported, because what you don't find in the interstitial fluid is clotting proteins and whole blood cells, right? That means it must have some degree of control, right? What you do find in the interstitial fluid, for instance, if I had glucose from what I ate in the blood, and I needed to get it into the interstitial fluid to ultimately get it to a cell, right, it would have to be able to travel, right? So this has some degree of control over the permeability of what's actually getting in. Does that make sense? Okay, so you probably remember that some capillaries are really porous and some capillaries are not very porous. So there are greater and lesser degrees of control. For instance, in the blood-brain barrier, it's really specific about what it lets through. But it has to let glucose through, right? It has to let oxygen through, right? Just doesn't let much of anything else. And then there are other places like huge sinusoid capillaries in the liver that are supposed to let just anything through in the whole cells. Does that make sense? So this can have different levels of porosity depending on where it is. Okay, so let's say we got something from the blood plasma into the interstitial fluid. So how would I get something between the extracellular fluid and the intracellular fluid, whether it was interstitial or blood plasma, or interstitial or blood plasma? So same question. What anatomical barrier is between the ECF and the ICF? Yeah, that's the cell membrane. That's what you were talking about. That makes sense? Now, I'm going to tell you for the 35th time in your academic career that the cell membrane has selective permeability, right? You know that. You've heard it a million times. What does that mean? It means the cell membrane is not willy-nilly about what it lets out. Like, you're not going to accidentally lose a nucleus, right, from the cell, right? So it has selective permeability about what it's going to transport. And the physiology will be determining the selective nature, right? What are you going to transport? When are you going to transport it? How much are you going to transport? And what would happen if you didn't? Does that make sense to you? Okay, so we're going to do a lot of talking about moving things around in the body. Okay, so these are your body fluid compartments that we've been talking about here. We've got the ICF and the ECF. Now, what kinds of things do you move around? The kinds of things that you move around, the first three are going to be obvious and you will have thought about them before. Obviously, you move around nutrients, right? And you move around gases, yes? Oxygen and carbon dioxide and glucose and amino acids and those kinds of things. And then, of course, you move around waste products, right? You have to get the waste products to the kidney or the liver or the digestive tract to get rid of them. But then the one that people are less familiar with and end up being a really important part of the phys story is ions, which if you want to sell something, you call them electrolytes. But they are ions, right? So if you're drinking your Gatorade, it's like, it's got electrolytes. It's basically sodium ions and potassium ions, right? That's what primarily it has in there in addition to fluid and glucose. Now, you might not at this point understand the importance of moving ions around the body, but we get a lot of that in phys. Almost the entire semester we talk about moving ions around in the body. And the fact that ions are charged, which you know from chemistry, ends up making the body have some charged electrical properties because the ions are charged. And I know that you have heard many, many times before that the nervous system is electrical. It's electrical because it moves massive amounts of ions from one place to another really quickly, which creates a big electrical flow. Right? So the ions and the electrolytes are going to be really important in understanding the electrical nature of the body. And it will seem a little hard to get at first, but eventually it'll start getting in there as well. So all of this moving stuff around is kind of creating an interesting sort of paradox, because what has to happen is the ECF has to be different from the ICF, because the ICF has different stuff in it, right? It's got cell stuff in it, right? And the ECF doesn't. So you have to be able to allow for exchange, but it has to be very controlled exchange. And the same thing here, blood plasma and interstitial fluid. And when you end up wanting exchange, but you want to keep things relatively constant, then you have to put in some money to keep things relatively constant to maintain homeostasis. It's just like at your house. If you want the inside of your house to be at 75, but it's 20 degrees outside and you got kids running in and out, in and out, and in and out, you're going to spend some money trying to maintain the homeostasis of your house, right? So the body spends about 60% of every cheeseburger that you eat simply regulating the composition, volume, temperature, and pressure of these fluids. Tremendous amount to maintain homeostasis. So I have a concept check question for you, and let's see if you can hazard a guess as to why that is. So if I told you that the body primarily assesses and tries to regulate very carefully one of these two types of fluid. So the body either spends a lot of time and money regulating ICF or a lot of time and money regulating ECF. Which one would you guess that it actually looks at most carefully? ICF? Anyone else? It's actually ECF. And I understand why you would think that, but I can lead you to understanding why this might be a better plan. First off, 100 trillion individual pools, right? Not one big pool. That sounds like a regulatory nightmare to me, managing 100 trillion individual pools. This one is one, or arguably two, big circulating pool. Do you see that it's a circulating pool because of the blood plasma, right? So what if instead of putting a temperature receptor inside every cell, I spaced them throughout the blood vessels so that they were actually assessing the body temperature as you go through? Right? Can you see that that would be more effective? In addition, I want you to think about this other idea. What if I took in a toxin? I ate something that was actually toxic. Question for you. Which fluid would it hit first, ECF or ICF? It would hit ECF first, right? And what if I took care of it before it got to the ICF and ended up changing cell function? Can you see that it's both more efficient to regulate the ECF and also safer to regulate the ECF? Does that make sense to you guys? So I'm not saying that each cell doesn't have like a means of regulating for itself, but I'm saying the entire body feedback. For instance, if your body temperature got too hot, it would be detected in a blood vessel, and you would tell the brain, and then the brain would say, okay, we actually need to vasodilate to the surface and activate our sweat glands and those kinds of things. I wouldn't wait for it to happen to every cell. Is that good? Okay, so because if the composition of the ECF changes very much, then the composition of the ICF is going to change, right? And if the composition of the ICF changes, then cell function is compromised, right? And then instead of being in a normal physiology situation, you're in a pathophysiology situation. And ideally, you would like to prevent that, if you could. Okay, so now, do we know everything about physiology? Absolutely not. Physiology is still being studied. What do we know? We have a really, really strong framework about how each body system works, but have we discovered any new neurotransmitters in the last 10 years? Yeah, we totally have. Have we discovered any new hormones? Have we discovered new functionality? So yeah, we're still understanding new functionality. And what I will commit to do for you is I try to stay as up on it as I possibly can because I love this stuff. And I will try to keep your information as current and relevant as I possibly can do it. So we will stop there for today because that's where we stopped in the last class, and also because it's a freaking monsoon outside. And if you have questions, you're welcome to email me or come talk to me. I already took roll. Did I take roll? Okay. I don't know why I finished early in your class.